Hello, guys, and welcome to the last part of our discussion about microbiology. So, let's get started with the diagnostic test for Enterobacteriaceae. Our first test is the oxidase test, and this test uses p-phenylenediamine dihydrochloride, and the positive result here is the formation of bluish purple color, and the negative reaction is no color change. So, Pseudomonas aeruginosa is positive for this test, whereas E. coli is negative for the oxidase test. Our next test is our nitrate reduction test, which uses sulfonylic acid and alpha-naphthylamide, and the positive color here is red and negative is colorless. So if it's negative, we should add zinc dust, and if it's positive, it should be colorless, and the negative reaction is red. As you can see in this picture, reduction to nitrate shows the positive color is red, whereas no reduction shows colorless, or the tube is colorless or doesn't have any color change. So for our next test, we have the orthonitrophenyl beta D galactopyranoside test. And this test detects the beta galactosidase, which converts on ONPG to orthonitrophenol. And the positive result here is yellow. And organisms who are positive are Shigella sonnei, Hafnia, and Yersinia. And the negative result here shows no color development. And the it shows or seen in other Shigella species, Salmonella, Proteus, Providentia, Edwardsiella, Erwinia, and Morganella. Next test is we have our LOE test, and this test detects decarboxylase and dihydrolase enzymes. So we have here our three amino acid that we are using and their corresponding end products. So for lysine, its end product is cadaverine; ornithine, its end product is putrescine; and arginine, its end product is citrulline. So take note that lysine or ornithine are used for the detection of decarboxylation, and for arginine, it's used for dihydrolation or to detect dihydrolation. So the medium used here is the molar decarboxylase base medium, and the positive result here is purple as seen in the first tube, and yellow if it's negative as seen in the second tube. And our third tube is used as a control as the uninoculated tube. So for the next test is we have our carbohydrate fermentation test, and it uses TSI or triple sugar iron dispense as length, and it has sucrose and lactose and but glucose, and it has glucose, and the ratio for the sugar are 10 is to 10 is to 1. So our pH indicator here is phenol red. So if it's acid, it will become yellow, and if it's alkaline, it will become red. And our H2S indicator here is ferrous sulfate, and our sulfur source is sodium thiosulfate. So bubbles or splitting of the medium in the butt or complete displacement of the medium from the bottom of the tube shows that an organism can produce gas or is able to exhibit gas production. So here's an example of a TSI tubes and their corresponding reaction. So for the interpretation of results for non-fermenter, it should be K over K or red over red. So no sugar was fermented. So for non-lactose fermenter, it's K over A or red over yellow, as seen in tube number 2. That means only glucose was fermented, and lactose fermenter shows A over A or yellow over a yellow, and 2 to 3 sugars were fermented as seen in tube number 1. So for the possible reactions, we have A over A can produce gas, and H2S negative can be seen in E. coli, Klebsiella, and Enterobacter. And our next reaction is K over A gas positive, H2S positive as seen in Salmonella, Proteus, and Edwardsiella. And our third reaction is K over A gas negative, H2S negative as seen in Shigella. And our last reaction is K over A gas negative, H2S negative as seen in Pseudomonas species. So for our next test, we have the lysine iron agar test, or the LIA, and the pH indicator here is our Bromcresol purple. Still, our H2S indicator here is our ferric ammonium citrate, or the H2S indicator is the ferric ammonium citrate, and our sulfur source still is the sodium thiosulfate. So for the lysine iron agar, it detects the amination that can be seen in the slant, and the positive color here is red or burgundy, and we use R, or we use the letter R for it. And if it's negative, it's purple or K. So the decarboxylation rather appears in the bot, and the positive or the positive result or color here is purple, and it's represented by the letter K, and the negative reaction is yellow. Our negative color is yellow and represented by the letter A. So the possible reaction here is R over A, which can be seen in Proteus, Providentia, and Morganella. K over K, which can be seen in Klebsiella, E. coli, Enterobacter, Edwardsiella, and Salmonella, and K over A that can be seen in Shigella. Here's the example of the LIA tubes with different reactions. So now let's proceed to the Invictus. So the Invictus is composed of indole test, methyl red test, Voges-Proskauer test, and citrate utilization test. So let's discuss them one by one. Let's start with the indole test. So this test is based on the ability of the organism to produce indole from tryptophan, and we use SIM or tryptophan broth for this test. And our agents used here are the COVAX or Ehrlich's reagent, or PDAB. And the positive result for the indole is red ring, and the negative result is no color development. Next test is we have our methyl red test, and this test detects the production of sufficient acid during the fermentation of glucose. So the medium used here is MRVP broth or Clark Lubes broth, and we use our methyl red indicator here, and the positive reaction is the distinct red color with pH less than 4.5, and if it's negative, we have no color change. Let's now move on to our VP test, or our Voges-Proskauer test. And this test detects acetone or acetyl carbonyl, and as we use in methyl red test, the MRVP broth or Clark-Lubes broth. And we have two methods with the Voges-Proskauer test. We have our Barrett's method, wherein we use alpha naphthol plus KOH, and the Coblin's method, where we use the alpha naphthol plus KOH plus creatine. So for the VP test, or for the Voges-Proskauer test, we have or pink to red color as the positive reaction, and no color change is negative reaction. And for the last test, we have the citrate utilization test, which is the C in the INVIC. So this is based on the ability of the organism to utilize citrate as a sole source of carbon. So we use Simon's citrate agar, and we use bromthymol blue as our indicator. So for the positive result, we have the intense blue color of the agar, and if it's negative, it's green. So here is the reaction, or here is the following Invictus and their positive and negative reactions. So for the first one, we have indole test showing pink ring here as our positive reaction, and for the methyl red, we have the distinct red color here, or yeah, distinct red color at the pH less than 4.5. And for the VP, it's pink or red color. And for citrate, we have blue as the positive color. So here, I summarize it for you guys, the different INVIC reaction this of the following members of Enterobacteriaceae. So when someone asks you what's the INVIC reaction of E. coli, you shouldn't answer like positive alone. So it should be complete. So for example, E. coli is positive, positive, negative, negative. And I also incorporated the TSI and LAA reaction of them. So please take note, guys, and the common ones here are the E. coli, which is positive, positive, negative, negative, Klebsiella pneumoniae, Enterobacter aerogenes, and Enterobacter cloacae, which is negative, negative, positive, positive. Late lactose fermenters, the Arizona, Citrobacter, Fungi, and Citrobacter diversus. We have the, or for Arizona, we have negative, positive, negative, positive. That's how you do an IMVIC. That's how you say an IMVIC reaction. It's just not positive alone. It should be complete. What's the reaction in indole test? What's the reaction in methyl red test? What's the reaction with Volks-Prescauer? What's the reaction with the citrate utilization test? So it should be complete. So please also take note, guys, the TSI and the LIA reaction of different organism or different members of Enterobacteriaceae, because this one is very important. So for our next test, we have the acetamide utilization test. And this test has the same principle with citrus test. And the positive color here is blue, which can be seen in Pseudomonas aeruginosa. And the negative reaction is no color change or development of a yellow color that can be seen in Stenotrophomonas maltophila. And our next test, we have the urease test. And this test is based on the conversion of ammonia through the action of urease. So the medium used here is the Stewart's urea broth or the Christensen's urea agar. So our indicator here is the phenol red. So for the positive reaction, it's the formation of magenta or bright pink color that can be seen in ppm for Proteus, Providentia, and Morganella, and negative shows no color change. Our next test is our esquilene hydrolysis test, and the indicator used here is the ferric ammonium citrate. And this test is presumptive for the identification of Klebsiella pneumoniae that is seen due to the black formation or development of black color in this medium. So our next test is our gelatin hydrolysis test, and this test shows that if the yes if the positive if the gel liquefies, it means that the reaction or the gelatin hydrolysis test is positive, and if the gel solidifies, it means that the gelatin hydrolysis test is negative. So if the gel liquefies or if it's positive, it can be seen in organisms like Proteus vulgaris and Serratia. And if the gel solidifies, indicating a negative reaction, it can be seen in Enterobacter aerogenes. So now let's proceed to the diagnostic test for Vibrio species. So for the string test or for our first test, we have our string test, and this test is used to separate Vibrio species from Aeromonas and Plesiomonas shigulloides. And this test is also used to differentiate Vibrio cholerae, which is string test positive, from other Vibrio species, which is string test negative. So for the positive result here, we have the appearance of string when the loop is pulled upwards for Vibrio cholerae. And if there's no appearance of string, it's negative and can be seen on Aeromonas species and Plesiomonas shigulloides. And also, another diagnostic test for Vibrio species is growth in TCBS agar. So we have here the table here shows the different microorganisms and their following characteristics. So for Vibrio cholerae, it has flat yellow colonies and 2 to 3 mm in diameter in TCBS, while Vibrio alginolyticus has large yellow colonies. Vibrio fluvialis and Vibrio vulnificus has yellow or translucent colonies. Vibrio parahemolyticus has colorless colonies with a green center, and Pseudomonas and Aeromonas has blue colonies, and Enterobacteria or others has tiny transparent colonies. So here is a picture of Vibrio cholerae and Vibrio parahemolyticus growth on TCBS agar. Next is we are going to learn the different diagnostic tests for non-fermentative organisms. So the OF test or the oxidative fermentative test, we use the media U and Leifson, and the indicator used here are the brom or is the bromthiamyl blue, and the positive reaction shows a yellow color, and if it's negative, it's blue or blue-green. And in the OF test, we inoculate by stabbing. So you can see in this picture that the first two tubes are the control. The second tube shows the growth of E. coli. The third, the third set of tubes shows the growth of Pseudomonas, and the last two tubes shows the growth of alkaligenous species. So for the OF tube, tube 1 is exposed to air, and this is considered as our open tube or our aerobic tube. And tube number 2 is overlaid with mineral oil. So that means this is our closed tube, and this is anaerobic. So if you're a fermentative organism, you should be positive to both open and closed tubes. And if you're an oxidative or oxidizer, you should be positive to open tube, but you should be negative to the closed tube. And if you're non-oxidizer or a saccharolytic, you should be negative to both open and closed tubes. So now let's proceed to the diagnostic test for acid-fast organisms. So as what we have learned in the part one of this lecture, acid fast staining is used to identify the organism up to the genus level only. And we have two ways how to report the AFB. We use the Center for Disease Control and the National Standard for Reporting Scale by the RITM, or used by the RITM and the Department of Health. So I want you guys to take note of their difference and how they report AFB scene and how do they grade each AFB, or how they grade AFB scene per visual fields. So next we have our fluorochrome staining. So this staining allows examination under LPO, and it's easier to read or leading for us to identify the organism easily. So the appearance of AFB under fluorochrome staining is yellow against a black background. So the primary stain here used here is the auramine rhodamine stain or the truant stain. Our decolorizer is still our acid alcohol, and for the counter stain, we use the potassium permanganate. So let's move on to our next test. And this test is called the niacin test. And this is useful in the identification of MTB because niacin is detected by aniline dye and cyanogen bromide. So the positive reaction here is yellow, as seen in the second tube, and it's presumptive for Mycobacterium tuberculosis, and negative shows no color change or colorless seen in Mycobacterium bovis. Now let's move on to the nitrate reduction test. And this test determines the production of an enzyme called nitrate reductase, which results in the reduction of nitrates. So the positive color here is red, and the organisms who are positive are Mycobacterium tuberculosis, Mycobacterium kansasii, and Mycobacterium avium complex, or MAC. Next, we will have our catalase test, and always remember that most mycobacterium species are catalase positive, but not all are heat stable catalase positive. So the media used here is the twin 80, and the reagent is the 30% H2O2 as what we use in our original catalase test. So the here is to heat, or we should heat the organism at 68 degrees Celsius for 20 minutes, and we should let it cool, then add the 30% H2O2. So for the positive result here, the height of the gas bubble should be greater than 45 millimeters as seen in Mycobacterium kansasii. And if the gas or if the height of the gas bubbles is less than 45 millimeters, it can be seen in Mycobacterium tuberculosis complex which consists of Mycobacterium tuberculosis, Mycobacterium bovis, Mycobacterium africanum, Mycobacterium ulcerans, and Mycobacterium leprae. Our next test is our iron uptake test, and this test is useful to distinguish Mycobacterium chelonae, which is negative, from other rapid growers which are positive. So the reagent used here is the 20% ferric ammonium citrate, and our positive result is the development of rusty brown color after adding the ferric ammonium citrate as seen in the picture. And our next test is our aryl sulfatase test. And this test is for the identification or for the presumptive identification of rapid growers. These are microbacterium that can grow in 2 to 3 days. And in this test, tripotassium phenolphthalein sulfate is a molecule from which phenolphthalein is liberated with exposure to aryl sulfatase. So for this test, the positive color is pink, which is for Mycobacterium chelonae, and negative result here is yellow, which can be seen in Mycobacterium fortuitum. And our next test is our hydrolysis of twin 80. So always remember that twin 80 is converted to oleic acid by lipase. So the pH indicator here is our neutral red. And this one or this test is useful in the identification of Mycobacterium cansaceae, which can grow on 3 to 6 days. And Mycobacterium tuberculosis is positive in 10 to 20 days. So the positive color here is pink or red, which can be seen on Mycobacterium marinum, Mycobacterium cansaceae, and Mycobacterium gordonei. And the negative result here is amber, and it's for non-photochromogenic mycobacteria. Our next test is the pyrazinamidase test, and this reaction usually occurs in about 4 days, and it is useful in distinguishing Mycobacterium marinum from Mycobacterium cansaceae and Mycobacterium bovis from Mycobacterium tuberculosis. So the positive color here is red, as seen in this first tube. Next test is we have our tellurite reduction test, and the reduction of colorless potassium tellurite to black metallic tellurium in 3 to 4 days. So the positive reaction here, of course, is black, which can be seen in Mycobacterium avium, and negative is colorless, which can be seen on Mycobacterium cansaceae. Next test is our urease test. This test, we use a loop full of test organism that is grown in 4 ml of urea broth at 37 degrees Celsius for 3 days. Our positive color here is red, which can be seen in Mycobacterium scrofulatium, and the negative here is yellow, which can be seen in Mycobacterium gordonei. Next test is we have the sodium chloride tolerance test. High salt concentration in egg-based media inhibits the growth of most mycobacteria. So the positive organisms here are Mycobacterium flavescens, Mycobacterium triviale, and Mycobacterium tuberculosis. Next, we have our thiophene 2 carboxylic acid hydrazide susceptibility test, or the T2H susceptibility test, and this test distinguishes Mycobacterium bovis, which is susceptible to T2H, from Mycobacterium tuberculosis, which is resistant to TOH, and also the growth on McConkey agar. And I want you to take note that Mycobacterium fortuitum and Mycobacterium chelonae can grow on McConkey agar. So also here, I made a table for the culture media for Mycobacterium. It's classified if it's non-selective media, agar-based, and liquid. So for the non-selective media or egg-based, we have here the egg-based media with malachite green. It inhibits contaminating organisms, and the growth is visible in 6 to 10 weeks. So we have 5 examples there. We have the Graf's modified algae media, the Lowenstein-Janssen, American Thoracic Society, LIS, Dorset egg medium, and Petrognani MINS. So for our agar base, we have our clear media for easy examination of colonies, and growth can be seen in 10 to 12 days, and these media can be used for pigment production and susceptibility testing. We have four examples rather. We have the Du Bois oleic acid media, the Mitchison's medium, the Middlebrook 7H10 with dextrose, and the Middlebrook 7H11 with casein hydrolysate. And the last one is we have our liquid media, and liquid media are non-conventional media, and growth is easily detected. So we have three examples there. We have the Bactec 12B, the Septicheck, and the Middlebrook 7H9. So I just want you guys to take note that LIS here means low inhibitory sterilized specimen, and MINS means maximum inhibitory and non-sterilized, or for non-sterilized specimen. Again, the American Thoracic LIS, or the LIS there, is low inhibitory for sterilized specimen, and Petrognani MINS there means maximum inhibitory for non-sterilized specimen. Now let's proceed to the diagnostic test for spirochetes. First, we have the diagnostic test for Treponema pallidum subspecies pallidum. And the methods of visualization here is we have two, the direct visualization that uses, or that can be, or we can use dark field microscopy, fluorescent antibody microscopy, or silver staining. And we also have serological tests that test for reagent and troponamal antibody test. Next, we have our Borrelia species, and the Borrelia species normally grows in a culture media called the Barber Stoner Kelly medium and Kelly's medium that is incubated at 33 degrees Celsius for 6 weeks. And also we use serological tests like ELISA and Western blot immunoassay. So diagnostic test for Leptospirus species. And for Leptospirus species, we use Fletcher's medium, EMGH medium, or the Ellinghausen Makolo Jensen Harris medium, and Noguchi. So for the serological test, we use MAT, or macroscopic agglutination test, and this one is a screening test. And number two, we have MIT, or the microscopic agglutination test, and this one is the confirmatory test. So now let's proceed for the diagnostic test for Chlamydia, Mycoplasma, Ureaplasma, and Rickettsiae. So for Chlamydia species, they do not grow on artificial culture media. They are often cultivated in tissue culture media like McCoy saline, Hella saline, and monkey kidney cell. So for Mycoplasma and Ureas plasma species, they require sterols for growth, and they are culturable in media and may produce colonies with fried egg appearance or mulberry colonies, as seen in this picture when stained with diane stain or methylene blue. So for to have the diagnostics test for each organism, we have first our Mycoplasma pneumoniae. So for the culture media, we have the PPLO agar, and PPLO here means pleuropneumonia-like organism agar. So for the test, we have the detection of cold agglutinin titer and produces cold agglutinins which reacts with anti-I. And we have the hemadsorption test, which can be used as the confirmatory test, and this test detects the ability to agglutinate guinea pig RBCs. And our last test is the growth inhibition test. So for Mycoplasma hominis, we require 7 to 8 days of incubation, and we use different culture media like A7A8 agar, NYC agar, and SP4 media with arginine. So for Ureaplasma urolyticum, culture media must be buffered to pH 6.0 due to growth inhibition by alkaline media. So we have here our culture media, still our A7, A8 agar, NYC agar, and you can see that the SP4 now is with urea as seen in this tube. So for Mycoplasma hominis, it's SP4 media with arginine as seen in this tube. And for Ureaplasma, it's SP4 with urea. So for our Rickettsia species, they do not grow on agar except for Bartonella. So rickettsial infections can be diagnosed by serological tests. And culture media used are the yolk sac of chick embryo or cell culture. And the special stains used, we have the Gemsa, the Machiavello, and the Jimenez. And now let's proceed to the diagnostic test for anaerobes. So always remember that the ideal specimen here for anaerobes is the needle aspiration of abscess, pus, and fluids. Swab is the least favorable method of collection. And if for urine, we use the suprapubic aspiration. And always remember that we should not refrigerate the specimen, and anaerobes require special nutritional requirements like vitamin K, hemin, yeast extract, and culture media use normally for the cultivation of anaerobes is we have the BBE or the Bacteroides bile esculin agar, the KVLB or the kanamycin vancomycin blood agar, the CCFA or the cefoxitin cycloserine fructus agar, the egg yolk agar, and the thioglycolate broth. So here we have the different medium. The first one is our BBE. The second one is our KVLB. And our next or our last agar here, our last medium here is the CCFA. So we have methods to promote anaerobiosis and storage requirements. So for anaerobic chambers, we have four requirements. So we have the catalyst, the desiccant, the oxygen reduction indicator, and the anaerobic gas. And we also have anaerobic jars, and we have PRAS or pre-reduced anaerobically sterilized medium. So we have here the different example. The first one is the anaerobic chamber. Number two is our anaerobic jar. And the next one or the last picture here is our press. So now let's proceed to the diagnostic test for fungi. So we have direct examination methods, namely the saline wet mount, the lactophenol cotton blue wet mount, the KOH preparation, the India ink staining, and the calcofluor white staining. So the first one, you can see that there's the lactophenol cotton blue. This picture is the India ink staining. This one is the KOH prep, and this one is the fluorescent staining or staining with calcofluor white. So for the diagnostic test, we also use culture media for the isolation and cultivation of fungi. So we have the Saborodextrose agar, we have the brain heart infusion agar, we have BHI enriched with blood and antibiotics, we have selected agars like inhibitory mold agar and dermatophyte test medium, and we also have differential agars, which are potato dextrose agar PDA, the birdseed agar, and the cornmeal agar with twin 80. So um these are the following culture media used for the identification of fungi. Okay, so now let's move on to the following tests are for the methods of yeast identification. So first we have our germ tube test. In this test, we use the zero point 0.5 ml and plus one colony, and we incubate for 2.5 to 3 hours. So for this test, we use zero point 0.5 ml serum or plasma plus one colony of yeast, and we incubate it for 2.5 to 3 hours. So the positive result here is germ tube production or elongated finger-like extensions from yeast cells. You can see it here. this one, this finger-like or this elongated finger-like extensions from yeast cells. That's the positive result for the germ tube test. So let me just go back here and emphasize this selective or this media. So for our first media here, we have our Saborid dextrose agar. And our next agar is this yellow. This one is the brain heart infusion agar. And this third media is the BHI enriched with blood and antibiotics. And it can be made selective by the addition of cyclohexamide, chloramphenicol, and gentamicin. So our last agar here, or our last medium here, is the inhibitory mold agar to recover Cryptococcus. And let's proceed. Our three tubes here, they are the dermatophyte test medium, mainly used for the isolation of trichophyton species. This one is the potato dextrose agar, and it's also used for the pigment of Trichophyton rubrum. But you can see here in this picture that Aspergillus flavus and Penicillium chrysogenum growth on PDA. So below here, the orange with black growth is the birdseed agar, and they are used for the recovery of Cryptococcus neoformans because black to brown colonies are due to the activity of phenyl oxidase. And the last media here is we have the cornmeal agar with twin 80, and this one can be used for the differentiation of Candida species, and it's useful for or to see or to better visualize the production of chlamydia spores. So now let's move on to the carbohydrate assimilation test. And this test identifies which carbohydrate or carbohydrates a yeast can utilize as a sole source of carbon. So for this test, the procedure is the freeze-dried sugar into wells, and we should add the yeast suspended in agar, and we incubate it at 30 degrees Celsius for 72 hours. So I made a table there, guys. So just familiarize yourself with the different Candida species and what carbohydrate can they assimilate. Next, we have our cornmeal agar morphology, and this one is to aid in the identification of pathogenic yeast by microscopic morphology. Candida albicans is used to demonstrate hyphae and chlamydospores as seen in this first picture, and Cryptococcus species is used to demonstrate blastoconidia as seen in the second picture. So next test is we have our potassium nitrate assimilation for the method of yeast identification. And this procedure provides valuable information for separating the clinically significant yeast. And the positive result here is it turns the medium blue, and the negative reaction area it turns the medium yellow. So control organisms that may be used for this test are Cryptococcus albidus, which is positive, and Candida albicans, which is negative. And the last test we have for yeast identification is we have our urease test. This test, our yeast isolates producing the enzyme urease can be detected easily with Christensen urea agar. So almost all clinically encountered Candida species are urease negative, and all Cryptococcus and Rhodotorella organisms are urease positive. Most strains, most strains rather of Trichosporin species are urease positive. So now let's proceed to the differential test for the dermatophytes. First, we have our hair perforation test. So for the procedure, we have 5 to 10 millimeters hair are floated on sterile water with few drops of sterile 10% yeast extracts. Then the conidia or hyphae are inoculated into water surface. And the last step is we have hair shafts are removed and examined in lactophenol cotton blue weekly for 1 month. So this test is presumptive for the identification of trichophyton mentagrophytes because it is hair perforation positive. Next step our next test rather is we have our 5-day urease test. So tubes of Christensen urea agar are lightly inoculated with dermatophyte and held for 5 days at room temperature. So P mentagrophytes or trichophyton mentagrophytes is 5-day urease positive. So another task is we have growth on rice grains. So here, sterile non-fortified rice is inoculated lightly with hyphae of isolate, then incubated at room temperature for 10 days, and then medium is observed for growth. So this test is presumptive for the identification of Microsporum canis. Now, let's proceed to the diagnostic test for viruses. So, specimen collection and transport. So, we have rules in specimen collection as well, with same with bacteriology and mycology. So, for the proper time, always collect the specimen early in the disease process, and also usage of appropriate means of transport, and you should always keep the specimen cool and moist. So for the transport medium, we have number one, the viral transport medium. And this one is consists of buffered isotonic solution plus protein with or without antibiotic, amphor and flextran. And swabs, we can use cotton, dacron, and nylon. So calcium alginate, always remember it's toxic and inhibits replication of some viruses and interferes with NAT. So tissues can be put on saline or TSB and must be kept moist from all the time. So for the container should be able to withstand freezing and thawing, and if delayed, it can be refrigerated or on ice, typically 4 to 6 degrees Celsius. So all specimen can be refrigerated except for blood, and blood specimen for CMV or respiratory specimen for RSV should not be frozen. So CMV here is cytomegalovirus, and RSV is our respiratory syncytial virus. So the next one, we have our processing and transport. So if processing is delayed 4 to 7 days, the specimen should be frozen and held at negative 70 degrees Celsius. So specimen should never be stored at negative 20 degrees Celsius because this temperature facilitates the formation of ice crystals. And the next one, proper specimen, go where the action is. So always remember that area involved is most likely to reveal the etiologic agent. So we also have three major ways in diagnostic or we use in diagnostic virology. One is direct detection, wherein we use immunofluorescence, EIA or ELISA, electron microscopy, immunoperoxidase staining, latex agglutination, radioimmunoassay, and nucleic acid probe assays and amplification methods. And we can also use serological testing that detects the viral antibody, and culture, which is the most versatile and comprehensive. In this culture, we commonly use tube culture or shell vial, and diagnosis is made within a day. And culture also enhances viral antigen detection tests. So we have three traditional methods for isolation of viruses. Namely, they are cell culture, which is most commonly used, animal inoculation, and use of embryonated eggs. So let's categorize the cell culture. The first one is our primary cell culture, and this is obtained from tissue removed from an animal. And our example here is the primary monkey kidney or PMK, and it's the most commonly used primary cell culture as seen in this picture. Our next one is the diploid cell cultures. These cell cultures are finite and can divide, but passage is limited to 50 generation. They contain 2 copies of each chromosome. So human neonatal lung or HNL is our standard here. And we have commercially available MRC5, which can be seen on the first picture, Y38 on the second picture, and the foreskin fibroblasts, which can be seen on the third picture. And next one, we have our continuous or heteroploid cell cultures. These cell cultures are capable of indefinite passage, or they are immortal cells. So we have here the different cell cultures and where are they derived from. So the HEP2 is derived from human laryngeal carcinoma. A549 is from human lung carcinoma. Vero is from monkey kidney. Hella is from cervical carcinoma. And KB is carcinoma of nasopharynx rather. So we have here the different cell cultures for the um, continuous or heteroploid cell cultures. So we have HEP2, we have A449, Vero, Hella, and KB. So our last line or our last in the category is the hybrid cell lines, and this is a mixture of two cell types, and this is a mix of A549 and MICLANG that support the growth of a wide range of viruses, namely the influenza, the parainfluenza, and adenoviruses. So that's all, guys, for microbiology. Thank you so much for listening, and I hope you learned something from me. Have a nice day.